Okay, so this is a seminar about PanBARLEX, a new web resource which will give us access to the latest iteration of the Barley Pan-genome. And it's a great pleasure to introduce Patrick König here today. So Patrick is a research software engineer at the IPK in Gatersleben in Germany. He studied hydrology at Dresden University and graduated from there in 2014. He then worked as a freelancer in web development until 2017, which is when he joined Uwe Scholz's group at IPK in Gatersleben, which is the Bioinformatics and Information Technology group. And he's been working there as a research software engineer ever since. So Patrick's been the main developer on projects like the Bridge project, which is a really information-rich digital front end to the IPK's huge barley germplasm collections. And if you work on barley, this is a great place to go and have a look at it. We've got links to all these resources in the appointment email that went out with the seminar. Patrick was also the main developer on DivBrowse, which is, again, a really rich resource on variomics. It's a web tool that allows you to interactively explore huge variant sets. So again, link to that is in your invite, and I would recommend you go and check it out if you don't know it already. So now Patrick is working on the SHAPE3 project, which is the continuation of the current Barley Pan-genome effort. And this is more focused on structural variation and haplotype diversity in the Barley Pan-genome. And I'm really looking forward to seeing the tools that come out of this. So he's also a co-lead in the Fair Agro Consortium, which is a German consortium that aims to develop tools and resources for fair data sharing. So just a couple of housekeeping things. Again, as per usual, the meeting's being recorded. So if you don't want your video to be recorded, then please turn your camera off. Please make sure you're muted at all times unless you're asking a question live. And if you've got questions, feel free to pop them into the chat during the talk, and then we'll go through them at the end. But there will also be lots of time to ask live questions after the presentation and to have a really good discussion. So I think with that, I can now hand over to Patrick. And
okay, yeah, thank you very much for this kind introduction of my person. Let me start the screen sharing, and so I hope now everyone can see my slides. Can you give me a...
Yeah, looks good. We can see them.
Okay, cool, thank you. Okay, hello everyone, and I want to tell you something about the new tool PanBARLEX, which we have developed in the frame of the Barley Pan-genome project. And so our motivation to develop this tool was that the second version of the Barley Pan-genome was released in February this year. Currently, it's available as a preprint on bioRxiv. And the version 2 of the Barley Pan-genome consists of 76 genotypes. And there roughly 3.8 million genes have been annotated. And also we have roughly 23 million functional annotations for all of these genes. And the whole data set of the Barley Pan-genome version 2 is spread across more than 600 files, and also different file types like GFF3, FASTA files, tabular, tab-separated value files. So it's, yeah, it's a bit, not kind of, a little bit kind of a mess to get into this data and to get an overview of this data. So our idea was then to develop a web tool for easy access to an integrated data set that makes life easier for every barley researcher, and which can help to gain insights into the Barley Pan-genome. So, and as a short recapitulation about the history of web tools about barley. So for the Morex version 3 reference, this reference was added, and also the gene annotation was added to the already existing PanBARLEX tool. And unfortunately for the Barley Pan-genome version 1, no interactive database or web tool was developed or is available to my knowledge. And now for the version 2 of the Barley Pan-genome, PanBARLEX is there, and yeah, we are looking forward if it can help. And here I want to show you a bit about the amount of data in the Barley Pan-genome. So we have, as I said before, more than 600 binary files or text files with content, and we have comma-separated value files, for example, with the functional annotations. We have the FASTA files with sequences and many more files of different types. And the approach in PanBARLEX was to integrate all those data sets into one relational database management system, or into a schema of relational database tables. And then a database and the web server was developed who deploys all this data via a REST API to a graphical user interface that can then be used by the users. Here's an overview about the functional annotations of the genes. And so all of the genes have annotations, but not in all categories of functional annotations. And as you can see here, for example, all of the genes have a human readable description for functional annotation, but we have only roughly 10,000 unique values. That means, for example, we have a lot of functional annotations with the value unknown protein, and these are occurring very often, and this reduces at the end the value of unique values. And so as you can see here, the most the highest amount of unique values is available with the TREMBL annotation. And yeah, if you are interested in the numbers, you can spot them later. Also, besides the gene annotation, there was analysis done with the AutoFinder tool by colleagues from Helmholtz Munich Center. It's the research group of Klaus Meyer and Manuel Spannagel. And they applied the AutoFinder tool to infer phylogenetic based autogroups. And here the tool originally was invented to apply it to compare different species, but now the scientific community also started to apply the tool to genotypes of one single species. And it's important to say that if there are artifacts in the gene annotation, those artifacts are also propagated into the downstream analysis with AutoFinder. And this can lead to some strange autogroups, which looking very strange. But we have checked the data set, and we also saw very reasonable looking results for some well-known genes. But at the end, it's important that all users who are use PanBARLEX must use the data carefully and always with a pinch of a healthy scientific skepticism, and don't take the data as a face value. So coming to the features of PanBARLEX, here you can see the initial start screen or the start page. And on this page, we have the main navigation on top. And here we also integrated a direct entry point for gene IDs and transcript IDs. We have integrated a search for functional annotations. This is a full text search. So where you can enter terms like kinase or some Go annotations or Go numbers, gene ontology numbers or IDs. We have here integrated, it's a blast search. We called it here homology search, but it's nothing else than a blast. And also we have here a link to the liftover from Morex version 3 gene IDs to the new Pan-genome gene IDs. So this is always a field for confusion, because between all the Morex reference genomes, we have we got new gene IDs, and even in the new Barley Pan-genome version 2 dataset, we have even another new version of the gene IDs. And because of this, we provide this liftover tool, which is based on a all versus all blast of the coding sequences. And it can help you to find the Morex version 3 genes in the Barley Pan-genome dataset. Also integrated is a direct access to Orthogroups. And here we have links to the Orthogroups tabular listing. And these are presets for Orthogroups, which are single copy core genome genes, multi-copy core genome genes, shell genome genes, and cloud genome genes. So this is more or less just a shortcut for the filter setting on the tabular listing of Orthogroups. Okay. It's now on. Okay, now it should work again. Okay, and on the bottom of the start page, we have some statistics, updates, and also references. Here's a screenshot of the gene details page. And as you can see here, on top, you get a short overview of the physical position of this gene on the chromosome of this genotype. And you got the functional annotations here listed, and also the specific features of this gene and sub-features like mRNA and exons. And when you click here on show sequences, it shows you the sequences, and you can just copy them to your clipboard and paste them somewhere else. Then we have here a screenshot of the tabular listing of all the 95,000 Orthogroups. And here you can directly see how many genes and genotypes are inside of this Orthogroup. You can see a functional annotation or multiple functional annotations of all the genes in this Orthogroup. And also you got here a visual representation of the spatial distribution of the genes across all chromosomes. So here you can directly see where the genes are located on the chromosomes. And this table can be filtered by functional annotations. Okay, fortunately, I got a message that my network connection is not stable. Is everything okay?
I can hear you okay, Patrick. Yeah, we can't see our slides now, but
yeah, I switched off. Now you can see the slides again, yeah?
Yeah, we're all good. We're all good.
Okay, cool. Yeah, this table of orthogroups can be filtered by different aspects, for example, by functional annotations, or you can say, I just want to get orthogroups where the genes are located on specific chromosomes. And also you can limit or filter the orthogroups by the number of genes and by the number of genotypes. And also, so with this filter setting, you can limit the orthogroups, for example, to single copy core genome genes or shell genome. So you can also use this presets here, which then fill the numbers here to get core genome, shell genome, and cloud genome genes. Yeah, this is what I said, just visualized with different colors. Then we have also a detail page for the orthogroup where you can see the visualization of the spatial distribution of genes in a bigger size, and also you can see below you get a table where you can see for all the 76 pan-genome genotypes which genes are in the orthogroup and also how many genes. And here in this example, we have the gene BRS1, and this gene is well known, and it controls the row type of barley. And if the gene is present, then the row type is 2-row, and if the gene is absent, then the row type is 6-row. And here you can see this, for example, Morex is a 6-row phenotype, and this corresponds to what we know. And also here you can see, for example, that the genotype HID 055 has this gene on another chromosome than all the other genotypes. So this list here is just a snapshot of all the 76 genotypes. But to illustrate how this works, I used also just this slice here of these genotypes. Because all 76 genotypes, the list or the table is too big to fit here on one slide. Also, it's possible to download all of the sequences of all genes of an orthogroup in a multi-FASTA file. This is possible for the coding sequences, for the transcripts, and also for the protein sequences. And this can help you to create, for example, multiple sequence alignment for all the genes of an orthogroup. So this, for example, here, I can download all the sequences for VRS1, and then I can, for example, create a multiple sequence alignment to see details or to see what is going on regarding the sequences. We have also integrated an interactive pairwise sequence alignment with the Smith-Waterman algorithm. Here you can enter or you can select a gene from your bookmark genes. So it's possible to create bookmarks for all genes. And then you can also change some parameters. And then you get the result. And for example, here in this example, you can see a single nucleotide variant where one base pair is different. Also, we have integrated SNPs from a VCF file, which is also part of the latest Barley Pan-genome paper. And this VCF file consists roughly 150 million SNPs for 1,315 genotypes. And this VCF file is referenced to the Murex genotype. And this is the reason why you can you get only the link to inspect the variants if you are on a Murex gene. So if you are on Murex, then you can click the link here, inspect variants in DivBrowse, and then DivBrowse opens in the same window and is directly the gene where you come from is directly visible in DivBrowse, and you can directly inspect the allelic diversity on a SNP level and also on a level of indels. We have a simple URL pattern to the landing pages of genes and orthogroups. As you can see here, it's always that the ID of the gene is added to a base path. And this is useful to send links around or to create links just based on the IDs. So if you work with the GFF3 files, for example, you can create those links automatically, for example. And that's the same for the orthogroups. Now I want to show you somewhat of a case study. Recently, a paper was published about the biosynthesis of gramine in barley. And the researchers or colleagues found out that the production of gramine is mainly controlled by a single gene. And this gene is cytochrome P450 monooxygenase, and then CYP76M5. This gene is located on chromosome 1H. And this study was conducted on the basis of the Barley Pan-genome version 1 with the 20 chromosome scale assemblies. And now I want to show you what's possible with PanBARLEX. So you can check the distribution and presence-absence variation of this gene for all the 76 genotypes. And to do so, I downloaded the coding sequence of the gene from NCBI GenBank. And the accession number, as you can see here, can be found in the section Data and Materials available of the paper if you want to try it also. And then I copied the coding sequence into the homology search window. I started the search, and then I got this BLAST result. And there I clicked on a gene of the first hits. And for example, here I clicked on the second one, which has a percentage of identical matches of 100%. And no mismatches in the alignment, so it could be a good hit. And when I get to the detailed page of this gene, I can see, for example, here in the annotations, there's also this cytochrome P450 annotation, and also I can see here this 76M5 annotation string. How to say, and even the physical position of the gene is on the same genomic region as stated in the publication about gramine. And now if I click then here on this Orthogroup ID link, I'm getting to the Orthogroup page of this gene. And as you can see here, all of the genes of this Orthogroup are located nearby on chromosome 1H in the same genomic region. And if I scroll down on this Orthogroup page, I can see the detailed presence-absence variation for this gene. And it's so that, for example, Barker and Bonus don't produce gramine because they don't have a copy of this gene. So now I want to give a disclaimer, and which we learned from past talks and discussions, which is in our feeling very important to give this disclaimer. And so the tool PanBARLEX only integrates and visualizes already existing or published Pan-genome version 2 data. So no further information is added. And the Barley Pan-genome version 2 dataset was generated collaboratively by various research groups contributing to the Barley Pan-genome project. And even we all know, I think, that gene annotation of genomes is still a challenging scientific domain. And therefore, we must be aware that according to the current state of the art, there can be no error-free gene annotation of a genome. So be aware, and annotation artifacts are to be expected, and don't take all the data as face value. And in general, we cannot guarantee that the data at all and the gene annotation and the grouping into orthogroups is 100% correct or 100% meaningful. Yeah. So little words about the reusability or possible reusability of the tool. So the software of PanBARLEX in principle can be reused for similar pan-genome datasets of other crops. And so if you run the AutoFinder tool on another crop, then yeah, in general it's possible. It's an amount of work to do so, but it's possible to spin up another instance which under another name, not PanBARLEX, because PanBARLEX is reserved to barley. But what you need for this would be then gene annotation in GFF3 file format, the AutoFinder output, then function annotations in CSV or tab-separated value files. The function annotations can be also integrated into the GFF3 file. And as additional input data, the FASTA files of the coding sequences and so on would be very useful to display and make it possible to download the sequences. And also a passport data, a table with passport data would be very useful. To summarize, the key aspects of PanBARLEX are: we have a fully integrated Barley Pan-genome version 2 dataset, and that allows easy exploration of the Barley Pan gene space. It provides convenient access to all genes and orthogroups. And we have a visualization of the spatial distribution and patterns of presence, absence, and copy number variation based on the derived orthogroups of AutoFinder. We have interactive tools like the interactive Smith Waterman alignment or pairwise sequence alignment, which helps to quickly analyze differences between a copy of genes. And it's possible to export sequences with few clicks. And also, as I said before, the software is in principle reusable for similar structured data set of other crops. And in future, our plans are regarding enhancements, are to link to grain genes for genome browsers. for the 76 chromosome scale assemblies. We are planning to integrate a web viewer for pre-calculated multiple sequence alignments of all these sequences of an orthogroup. So the user has to download the sequences and make the multiple sequence alignment on his own. He can do so if he wants to use a specific tool, but we think it's a good idea to provide pre-calculated multiple sequence alignment. Then a sequence export is planned where you can export any sequence of interest easily from all the 76 assemblies. A dot plot generator is planned where you can create interactive dot plots of the sequences in the web browser, and also we are currently checking the integration of predictions of protein structures of all the genes. And we are happy to receive your feedback or your ideas to improve the PanBARLEX portal to gain more usefulness for the Barley research community. And the tool is available under this URL. And so I'm coming to the end. I want to thank you for your attention, and I also want to thank all the Barley Pan-genome version 2 or the Barley Pan-genome project collaborators who made this web portal possible and who created all the data and did the sequencing and so on. So it's really a community project at the end that PanBARLEX is possible. And special thanks to the Plant Genome and Systems Biology group at Helmholtz Center Munich, around Thomas Lux, Manuel Spannagel, Heidrun Gundlach, and Klaus Meyer for the gene annotation and the AutoFinder analysis, and lots of discussions about this AutoFinder tool. And also special thanks to my colleagues at IPK, Uwe Scholz, Martin, Uwe, and Niels, for giving guidance and advices how to present such a tool like PanBARLEX. Okay, that was my presentation. Thank you very much, and thank you very much for the opportunity to present here in the frame of the Barley Hub or International Barley Seminar. Thank you very much.
Thank you very much, Patrick. That's great. It's been a really good detailed introduction into PanBARLEX, and I can see how lots of people here on the call will be excited to be using this in the future, because this is really a fantastic resource, and it's a good way of interrogating the pan-genome. So it's brilliant. So there's quite a lot of questions in the talk. I'll maybe go through these first, and then we can ask some live questions after that. So just taking it from the top, I've posted a link to the pan-genome paper into the chat if you haven't seen that. So feel free to go and check that out. So Laura was asking, are there any plans to add a reverse of the leftover from the MORXV3 to find the best matching MORXV3 gene from a pan-genome Orbit code? And Uwe's already provided an answer to that. So this is going to be explored, and will probably be done in conjunction with the colleagues from Munich. So then Robbie has posted a reminder that there's still vacant dates for further seminars if anyone wants to give a presentation. Dangping Liu is asking, can we search the orthologs from other species genomes? And again, Uwe's provided an answer saying this is not possible, but I'm not sure, Uwe, that this is what Dangping was asking. Dangping, if you're there, do you want to unmute and maybe clarify this? I think maybe you meant if you have a sequence from another species, can you search the barley orthologs in PanBARLEX with that? Is that what you meant? We still got Dangping there.
Patrick, could you
yes.
turn off your presentation mode, Patrick, so we can see people?
Yeah, actually, that would be good. Sorry.
I don't know where to stop the. Here.
There's a floating
Okay, now
menu
it's
somewhere
better.
on the script.
That's it?
On your screen, rather. Yeah, so I don't think we've got Dang Ping, are you there?
Yeah.
Do you want to clarify what you meant? Did you mean can you search PanBARLEX with a sequence from another species?
Yes, I just want to ask, can I search the other genome sequence? I just I'm interested with this tall. So I think can I search is also like use this tall to search also for other genome sequence. So that's my question. So I think it's not a problem. I think I already got the answers. Yeah.
Okay. Yeah, I think my impression was you can use the blast search to put any sequence in, and as long as it returns a hit against PanBARLEX, then you will have an answer.
Yeah. I got the answer. Yeah. Thank you.
Okay, thank you. So Runshan is saying any possibility of mismapping of the orthologs on the wrong chromosome. I think there were probably a few raised eyebrows with the VRS1 gene on chromosome 4. And it's either a really interesting new biological finding, or it could be a bioinformatics artifact. And I think, we all know that large-scale automatic annotation with bioinformatics tool is never perfect. And so, yeah, I think we need to definitely be open to that possibility. And there's a whole kind of discussion after that. So Robbie is saying, if I recall correctly, the VRS1 gene is present in Morix, but it contains a point mutation and knocks out gene function. Does this mean that most all genes with deleterious mutations that knock a coding sequence out of frame are missed from the database, or would it be detected and flagged as non-functional? Patrick, do you want to answer that?
There's a kind of answer subsequently in the chat, by the way.
Yeah, please. Yeah, I think this is really, it's a good question, but also very hard to answer. I think it could be the reason that for Morix there was no copy. It could be that theoretically the AutoFinder algorithm was not putting the already existing copy in the same auto group. We can check this. So yeah, I think this has to be checked. It's a very specific question, and we can check what's the reason why for Morix was no gene present in this auto group of VRS1. Yeah.
I think it'll be highly dependent on the parameters in AutoFinder, because if, as Robbie suggests, you have a deleterious mutation like an early stop codon, and your protein sequence is only half the length of what it should be in the author group, then AutoFinder might drop it from the author group altogether. That's likely what happens.
Yeah, sometimes
because
sometimes maybe Anna or Bruno could answer. There is an answer to that question in the text, in the chat.
Okay. Yeah, sometimes we have observed that some of the genes which should be in one auto group are split into 2 or 3 auto groups. I think this could be the reason of thresholds in the AutoFinder method. Yeah,
Anna is saying the same thing. So, and she's saying she's checked that previously. So that's okay. So that's fine. Then Carmen was asking for inspecting variants, reference Morix. What if our gene does not exist in the Morix genome?
Yeah, then it's a pity, because if this gene is not annotated in Morix, you cannot see it in the diff browse instance. So that's clearly a drawback of the reference genomes, or the approach of having SNP panels bound to a specific reference. Yeah, this is then not possible to see.
But we will have the gene in the Orthogroups, even if it's not in Barlex. So that's kind of, at least you'll be able to inspect it in the context of the Orthogroups. So that should be okay.
Yeah. As long as it is annotated in some of the, or in at least one of the 76 Barley Pan-genome genotypes, then you could find it in the Pan-genome, but not in the SNP browser, because this is bound to Morex genes.
Yeah.
Yes, thank you.
So a little bit more discussion about orthogroups there, but we've kind of covered that already. So Runshan is commenting that agree that no annotation is error-free, but some estimation of false discovery rate would be very helpful. Yeah, that's a good point. Do you want to answer that, Patrick or Uwe?
Yeah,
I suppose that's quite tricky because you know, what do you use as your truth set in this context?
so this is fits. I would try to answer or not to answer, only give a comment. So this is always a good question for this false discovery rates, and this fits to the answer what I wrote before, that we really start now the discussion with the people in Munich about such issues. Yeah, that I think with the tool it was possible to see this and to illustrate this. But this is always the background of science. You generate with new presentation, new results, more questions. And I think this is good, and we are going forward with this. So that's why a very good question. Up to now, no answer, but we will discuss the possibilities and try to put it to have it on the agenda.
Yeah, I apologies that I've been very tough on you guys. I know all the methods you guys applied is very state of art, but it does bother me. Many of the state of art methods, they have a lot of false positive rates, but they don't mention about it. I think it would be good for the community to know, for example, you know, we all accept nothing is perfect, but if you say like 10% of them could be problematic, or where the problematic G models could be, that would be extremely helpful for the community to say, well, for this G model is likely to be wrong because we know author finders used to have this kind of problems. But as you said, this is probably ongoing research has to be carried out. Thank you.
Yeah, thank you. So then a comment from Robbie at Patrick or Uwe. I would suggest that linking to a gene expression browser like Iona would be a valuable addition. And two people have given a thumbs up to that. I'm very much in favor of that too. If you want to do that, come and speak to myself or to Craig Simpson or Linda Milne. And that would be nice to have, you know, links going both ways. That would be really good.
I think we will do this, Michael.
Yeah. I mean, that's a fairly straightforward thing, probably just, you know, bouncing gene IDs back and forth, providing we can translate them suitably. This is an eternal problem that will really, you know, only get worse as time goes on, and we need to kind of think about how we deal with all the varying gene IDs in the different annotation sets. Robbie is also suggesting, Patrick, that you upload your talk to YouTube. Or actually, no, is this what we do, Robbie? Is that what you mean?
Yes.
Yeah, okay, we upload our talks to YouTube.
Yeah, you do that. Yeah, okay.
Yeah. And then that will help people with how to use it. So that's great. Uzal Litson, if I pronounced that correctly, says, thank you for the nice presentation. Are the PanBARLEX gene IDs user-friendly for other databases like ensemble plant or grain genes?
Okay, I don't know exactly what is meant with user-friendly, but for grain genes, it is that they use the same gene IDs because they use the same GFF3 files. I don't know what is going on Ensemble Plant. If they want to integrate the Barley Pan-genome data set, I don't know. I can't say about such plans. I don't know if anyone else maybe can say about Ensemble Plant.
I think currently Ensemble Plant only supports Morex v3. And like you, I don't know whether they've got any plans on supporting pan-genome IDs. I don't think they've been part of the consortium in any capacity on this occasion.
Okay, next question or?
Yeah, so can additional genomes be integrated easily, asks Klaus Pillen?
So this has to do with the data set. So if you want to add, for example, another genotype to the already existing 76 genotypes, you have to annotate, or first you have to create the assembly, then you have to annotate in the same way as you have annotated the previous 76 genotypes. And then you have to redo the calculation of AutoFinder to get a new data set based on the new ground or basement data set. And so this is a lot of computational effort. I think it's you cannot say cannot be easily done because it needs a lot of effort to add additional genomes. It's not super complex or complicated, but it's effort to do so. And then also you got a new version of the pan-genome. So if we say we have now the 76 genotypes pan-genome, if we add further genotypes, then we expand the pan-genome and we get a new version. So even we have then to think about versioning or different versions of the pan-genome, and everything gets more complex and maybe also confusing. This can just be answered from the whole community, how we proceed with the pan-genome. It implicates a lot of tackles.
Okay, so Martin has just answered our question about the VRS1 copy on 4H. So the accession with that which is HID 055, has a reciprocal 2H 4H translocation. So there you go. So there's your answer there. So that copy of VRS1 will probably be in the translocation. Okay, interesting. So not an artifact on this occasion, but be wary of artifacts always. Amy Learmonth is asking, will the new laureate genome be included in this? It's not, unfortunately. It was only Miriam only just finished it yesterday. And the same applies as what Klaus Pillen just asked. It would basically require for the whole of the pan-genome analysis to be repeated and the orthogroups to be redone. So I would imagine that at some point, given that we are now working on the Shape3 project, maybe when at the end of that, will we get a new version of the orthogroup analysis?
I think so, yeah.
That would make a great deal of sense, wouldn't it? Okay, so then Malcolm has posted a link to previous seminars. If you want to see those, then please go to the chat and look near the bottom. Dangping Lue, one more question. Has the RNA-seq data been used in the annotation?
To my knowledge, yes. It's also stated in the publication or in the manuscript. That the RNA-seq data of 20 genotypes has been used for de novo annotation, and for the rest of the genotypes, the 56 other genotypes, there was a so-called projection pipeline where the gene annotation, the de novo gene annotation was projected onto the 56 other genotypes without RNA-seq data.
Yeah. So
But
Duncan,
this is clearly, I think this is clearly stated in the manuscript.
yeah, it was also, there was also ISO-seq data, not just RNA-seq data. So, and this is essentially the data set which makes up the Barley Pan transcriptome, which is also about to get published. So, another comment from Robbie saying we have 3 vacant dates for further seminars in this series before Christmas 2024. Would anyone in the audience today like to volunteer to give a presentation or recommend someone that you would really like to hear give a presentation in this forum? Please add suggestions to the chat and we will take it from there. Okay, so here's another question from Klaus Pillen. Due to the wide collinearity of serial genomes, one could think of combining, for instance, a meta-pan-genome of barley, wheat, and rye. And that's got 1 thumbs up so far. Is that an option?
It was my thumb. So from a perspective as a tool developer who developed this PanBARLEX tool, it would not be a problem. But I think the experts on the genetic level, like Robbie and Mika and so on, and Niels, they should then say more about if it makes sense. And I think it's really an interesting option to do so. Yeah. But maybe another expert on this topic here can give a more qualified answer than me.
Yeah, actually, I mean, on this topic, so some people might be aware that there is a pan-Ordeum effort underway. And Robbie, I think that would actually be a real nice talk for the seminar series if it hasn't already been and I've missed it. You still there, Robbie? Yeah. So perhaps we can, I mean, this is related to that, and perhaps we can recruit someone from the pan-Ordeum consortium to give a presentation of where we're at, but that would probably be later in the year.
You could ask Martin directly, he's online.
Sorry, who?
Martin.
Martin, yeah. Martin or anyone else. Martin would probably be the obvious person. Martin, are you there? Do you want to volunteer? Shall we volunteer you?
Or volunteer somebody.
Or volunteer someone else, yeah. He was there a moment ago. Okay, so at the moment there's no more questions in the
So Martin has left
the building.
Yeah.
So there's no more questions in the chat at the moment. Would anyone like to unmute and ask a question, or maybe stick a hand up best because there's quite a few of us, and we'll see if there's anything we haven't covered yet. I'll have a look for raised hands. Don't see anyone at the moment. No, no one. Well, if we're all out of questions, then I think we can probably bring this to a conclusion. Patrick, I would like to thank you very much for doing this. This was a great talk. It'll be really useful for people learning how to use PanBARLEX. And I'm sure you wouldn't mind if people send you emails with questions, perhaps following on from today. And thank you to Uwe as well. And thank you to everyone who's attended today. And we will hopefully see you all at the next seminar.
Just
Thank
want
you.
to,
Oh,
okay,
sorry.
I just want to add as a latest or last statement, if you have specific questions to regarding the gene annotation and the autofinder analysis, then you can send your questions directly to the group of Klaus Meyer and Manuel Spannagelt and at Helmholtz Munich. So Manuel sent me an email before that I can say this. So if you have questions regarding the gene annotation please directly contact Manuel Spannagel and colleagues at Helmholtz Munich.
Yeah, that's great. That's a good point. Okay, so once again, thank you all for coming. Thank you, Patrick, for a great talk, and hopefully we'll see you all at the next seminar.
Thanks,
Bye,
Patrick.
Thank you.
Bye-bye. Thank you.
Suggestions for speakers, mail them to me.
Bye.
I can see some people online that I might suggest for speakers. Thanks, everybody.